Lesson 5, Fundamental Unit of Life. While examining a thin slice of cork, Robert Hooke saw that the cork resembled the structure of a honeycomb consisting of many little compartments. Cork is a substance which comes from the bark of a tree. This was in the year 1665 when Hooke made this chance observation through a self designed microscope. Robert Hooke called these boxes as cells. Cell is a Latin word for a little room. This may seem to be a very small and insignificant incident, but it is very important in the history of science. This was the very first time that someone had observed that living things appear to consist of separate units. The use of the word cell to describe these units is being used till this day in biology. Let us find about cell. What are living organisms made up of? Let us take a small piece of an onion bulb. With the help of a pair of forceps, we can peel off the skin called epidermis from the concave side, which is the inner layer of the onion. This layer can be put immediately in a watch glass containing water. This will prevent the peel from getting folded or getting dry. What do we do with this peel? Let us take a glass slide, put a drop of water on it, and transfer a small piece of peel from watch glass to the slide. Make sure that the peel is perfectly flat on the slide. A thin camel hair paintbrush might be necessary to help transfer the peel. Now we put a drop of saffron in solution on this piece, followed by a cover slip. Take care. Avoid air bubbles while putting the cover slip with the help of a mounting needle. Ask your teacher for help. We have prepared a temporary mount of onion peel. We can observe this slide under low power followed by high power of a compound microscope. What do we observe as we look through the lens? Can we draw the structures that we are able to see through the microscope on an observation sheet? Does it look like figure 5.2, which shows cells of an onion peel? We can try preparing temporary mounts of peels of onions of different sizes. What do we observe? Do we see similar structures on different structures? What are these structures? These structures look similar to each other. Together they form a big structure like an onion bulb. We find from this activity that onion bulbs of different sizes have similar small structures visible under a microscope. The cells of the onion peel will all look the same regardless of the size of the onion they came from. These small structures that we see are the basic building units of onion bulb. These structures are called cells. Not only onions but all organisms that we observe around are made up of cells. However, there are also single cells that live on their own. Cells were first discovered by Robert Hooke in 1665. He observed the cells in a cork slice with the help of a primitive microscope. Leeuwenhoek 1674 with the improved microscope discovered the free living cells in pond water for the first time. It was Robert Brown in 1831 who discovered the nucleus in the cell. Purkinje in 1839 coined the term protoplasm for the fluid substance of the cell. The cell theory that all the plants and animals are composed of cells and that the cell is the basic unit of life was presented by two biologists, Schleiden and Schwann. The cell theory was further expanded by Virchow 1855 by suggesting that all cells arise from pre-existing cells. With the discovery of the electron microscope in 1940, it was possible to observe and understand the complex structure of cell and its various organelles. The invention of magnifying lenses led to the discovery of microscopic world. It is Now known that a single cell may constitute a whole organism, as in amoeba, Chlamydomonas, Paramecium, and bacteria. These organisms are called unicellular organisms. On the other hand, many cells group together in a single body and assume different functions in it to form various body parts in multicellular organisms, such as some fungi, plants, and animals. Can we find out names of some more unicellular organisms? Every multicellular organism has come from a single cell. How? Cells divide to produce cells of their own kind. All cells thus come from pre existing cells. We can try preparing temporary mounds of leaf peels, tip of roots of onion, or even peels of onions of different sizes. After performing the above activity, let us see what the answers to the following questions would be. Do all cells look alike in terms of shape and size? Do all cells look alike in structure? Could we find differences among cells from different parts of a plant body? What similarities could we find? Some organisms can also have cells of different kinds. Look at the following picture. It depicts some cells from human body. The shape and size of cells are related to specific function they perform. Some cells like amoeba have changing shapes. In some cases, the cell shape could be more or less fixed and peculiar for a particular type of cell. For example, nerve cells have typical shape. Each living cell has the capacity to perform certain basic functions that are characteristic of all living organisms. How does a living cell perform these basic functions? We know that there is a division of labor in multicellular organisms such as human beings. This means that different parts of the human body perform different functions. The human body has a heart to pump blood, a stomach to digest food, and so on. Similarly, division of labor is also seen within a single a single cell. In fact, each cell has got certain specific components within it known as cell organelles. Each kind of cell organelle performs a special function, such as making new material in the cell, clearing up the waste material from the cell, and so on. A cell is able to live and perform all its functions because of these organelles. These organelles together constitute the basic unit called as cell. It is interesting that all cells are found to have the same organelles no matter what their function is or what organisms they are found in. What is a cell made up of? What is the structural organization of a cell? We saw above that the cell has special components called organelles. How is a cell organized? If we study a cell under a microscope, we would come across three features in almost all cells. Every cell. Plasma membrane, nucleus, cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these features. Let us see how. Plasma membrane or cell membrane. This is the outermost covering of the cell that separates the contents of the cell from its external environment. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. It also prevents movement of some other materials. The cell membrane, therefore, is called a selectively permeable membrane. How does the movement of substances take place into the cell? How do substances move out of the cell? Some substances like carbon dioxide or oxygen can move across the cell membrane by a process called as diffusion. We have studied the process of diffusion in earlier chapters. We saw that there is spontaneous movement of a substance from a region of high concentration to region where its concentration is low. Something similar to this happens in cells when, for example, some substance like carbon dioxide, which is cellular waste and requires to be excreted out by the cell, accumulates in high amount inside the cell. In the cell's external environment, the concentration of carbon dioxide is low as compared to that inside the cell. As soon as there is a difference of concentration of carbon dioxide inside and outside a cell, carbon dioxide moves out of the cell from a region of high concentration to a region of low concentration outside the cell by the process of diffusion. Similar oxygen enters the cell by the process of diffusion when the level of concentration of oxygen inside the cell decreases. Thus, diffusion plays an important role in gaseous exchange between the cells as well as the cell and its external environment. Water also obeys the law of diffusion. The movement of water molecules through such a selectively permeable membrane is called osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved in water. Thus, osmosis is the net diffusion of water across a selectively permeable membrane toward a higher solute concentration. What will happen if we put an animal cell or a plant cell into a solution of a sugar or salt in water? One of the following three things could happen. If the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis. Such a solution is known as hypotonic solution. Water molecules are free to pass across the cell membrane in both directions, but more water will come into the cell than will leave. The net overall result is that water enters the cell is likely to swell up. If the medium has exactly the same water concentration as the cell, there will be no net movement of water across the cell membrane. Such a solution is called as isotonic solution. Water crosses the cell membrane in both directions, but the amount going in is the same as the amount going out. So there is no overall movement of water. The cell will stay the same size. If the medium has a lower concentration of water than the cell, meaning that it is very concentrated solution, the cell will lose water by osmosis. Such a solution is known as hypertonic solution. Again, water crosses the cell membrane in both directions, but this time more water leaves the cell and then enters it. Therefore, the cell will shrink. Thus, osmosis is a special case of diffusion through a selectively permeable membrane. Now, let us try out the following activity. Activity: osmosis with an egg. Remove the shell of an egg by dissolving it in dilute hydrochloric acid. The shell is mostly calcium carbonate. A thin outer skin now encloses the egg. Put the egg in pure water and observe after 5 minutes. What do we observe? The egg swells because water passes into it by osmosis. Place a similar deshelled egg in a concentrated salt solution and observe for 5 minutes. The egg shrinks. Why? Water passes out of the egg solution into the salt solution because the salt solution is more concentrated. We can also try a similar activity with dried raisins or apricots. Activity: put dried raisins or apricots in plain water and leave them for some time. Then place them into a concentrated solution of sugar or salt. You will observe the following. Each gains water and swells when placed in water. However, when placed in concentrated solution, it loses water and shrinks. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis. Absorption of water by plant roots is also an example of osmosis. Thus, diffusion is important in exchange of gases and water in life of a cell. In addition to this, the cell also obtains nutrition from its environment. Different molecules move in and out of the cell through a type of transport requiring use of energy. The plasma membrane is flexible and is made up of organic molecules called lipids and proteins. However, we can observe the structure of the plasma membrane only through an electron microscope. The flexibility of the cell membrane also enables the cell to engulf in food and other material from its external environment. Such processes are known as endocytosis. Amoeba acquires its food through such processes. Activity: find out about electron microscopes from resources in the school library or through the internet. Discuss it with your teacher. Cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose. Cellulose is a complex substance and provides structural strength to plants. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. This phenomenon is known as plasmolysis. We can observe this phenomenon by performing the following activity. Mount the peel of a royal leaf in water on a slide and examine cells under the high power of a microscope. Note the small green granules called chloroplast. They contain a green substance called chlorophyll. Put a strong solution of sugar on salt on the mounted leaf on the slide. Wait for a minute and observe under a microscope. What do we see? Now place some royo leaves in boiling water for a few minutes. This kills the cells. Then mount one leaf on a slide and observe it under a microscope. Put a strong solution of sugar or salt on the mounted leaf on the slide. Wait for a minute and observe it again. What do we find? Did plasmolysis occur now? What do we infer from this activity? It appears that only living cells and not dead cells are able to absorb water by osmosis. Cell walls permit the cells of plants, fungi, and bacteria to withstand very dilute hypotonic external media without bursting. In such media, the cells tend to take up water by osmosis. The cell swells, building up pressure against the cell wall. The wall exerts an equal pressure against the swollen cell. Because of their walls, such cells can withstand much greater changes in the surrounding medium than animal cells. Nucleus. Remember the temporary mount of onion peel we prepared? We had put iodine solution on the peel. Why? What would we see if we tried observing the peel without putting the iodine solution? Try it and see what the difference is. Further, when we put iodine solution on the peel, did each cell get evenly colored? According to their chemical composition, different regions of cells get colored differently. Some regions appear darker than other regions. Apart from iodine solution, we could also use safranin solution or methylene blue solution to stain the cells. We have observed cells from an onion. Let us now observe cells from our own body. Activity. Let us take a glass slide with a drop of water on it. Using an ice cream spoon, gently scrape the inside surface of the cheek. Does any material get stuck on the spoon? With the help of a needle, we can transfer this material and spread it evenly on the glass slide kept ready for this. To color the material, we can put a drop of methylene blue solution on it. Now the material is ready for observation under microscope. Do not forget to put a cover slip on it. What do we observe? What is the shape of the cells we see? Draw it on the observation sheet. Was there a darkly colored spherical or oval dot-like structure near the center of each cell? This structure is called nucleus. Were there similar structures in onion peel cells? The nucleus has a double layered covering called nuclear membrane. The nuclear membrane has pores which allow the transfer of material from inside the nucleus to its outside, that is, to the cytoplasm. The nucleus contains chromosomes which are visible as rod-shaped structures only when the cell is about to divide. Chromosomes contain information for inheritance of characters from parents to next generation in the form of DNA, deoxyribonucleic acid molecules. Chromosomes are composed of DNA and protein. DNA molecules contain the information necessary for constructing and organizing cells. Functional segments of DNA are called genes. In a cell which is not dividing, this DNA is present as part of chromatin material. Chromatin material is visible as entangled mass of thread-like structures whenever the cell is about to divide. The chromatin material gets organized into chromosomes. The nucleus plays a central role in cellular reproduction, the process by which a single cell divides and forms two new cells. It also plays a crucial part along with the environment in determining the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of a nuclear membrane. In such an undefined nuclear region, containing only nucleic acid is called a nucleoid. Such organisms whose cells lack a nuclear membrane are called as prokaryotes or primitive nucleus. Organisms with cells having a nuclear membrane are called as eukaryotes. Prokaryotic cells also lack most of the other cytoplasmic organelles present in eukaryotic cells. Many of the functions of such organelles are also performed by poorly organized parts of the cytoplasm. The chlorophyll in photosynthetic prokaryotic bacteria is associated with membranous vesicles, bag like structures, but not with plastids as in eukaryotic cells. Cytoplasm. When we look at the temporary mount of onion